Exciting new research recently revealed the surprising role of the immune system in generating fibromyalgia pain. Not only does this information potentially vastly expand our treatment options, it also provided what I think was the final puzzle piece in understanding the chain reaction of events that cause fibromyalgia pain. I am really excited. I just had a journal article published about my findings and interpretation that shows in great detail the supporting evidence for my hypothesis about how to include this immune system data into fibromyalgia pain and how that can direct our treatment options going forward. In this video, I'm going to present the same information, but in a less sciencey, more understandable format, because I feel like so many of us with fibromyalgia are in that unenviable position of having a doctor that's not keeping up with what's going on in fibromyalgia. And we find ourselves in the position of needing to bring that information to our doctors. So with this video, I'm going to try to make sure that you understand everything that this new immune system data revealed about fibromyalgia pain. I'm Dr. Ginevra. I'm a physician with fibromyalgia myself. I'm on a mission to empower everyone with fibromyalgia with the knowledge and inspiration they need to defeat this illness. I call this being fibro-fierce. I'm so glad you've joined us. Let's dive into the science. First, we're going to take a look at what we already knew about the science of fibromyalgia pain prior to this mouse antibody study that showed that the immune system seems to play a big role in that process. And then I'm going to go into some detail so you can really understand what the mouse antibody study showed and didn't show about the relationship of the immune system to fibromyalgia pain. And then finally, I'm going to outline the hypothesis, the theory that I presented in my recently published journal article that incorporates this new immune system data into the chain reaction that we think causes fibromyalgia pain. So what did we already know prior to this new study? Well, we have a lot of research showing that there is central sensitization in fibromyalgia. That's a bit of a mouthful, but what it means is that the central nervous system, that's our brain and spinal cord, has become sensitized to pain signals so that all the pain signals that it's receiving, it's turning the volume up on those signals. And that is sort of an abnormal pain amplification process. We also know that in fibromyalgia, the sympathetic nervous system, that's the nervous system that controls the fight or flight or kind of danger response activities in the body. We know in fibromyalgia that there is significant hyperactivity, overactivity of the fight or flight nervous system. It should only be put into place when we actually are exposed to danger. But in fibromyalgia, there's a smoke alarm going off in our brain that continually is activating that fight or flight response. And that leads to a lot of the downstream effects or the symptoms that we see in fibromyalgia. The other thing that we know is happening in large part due to this sympathetic nervous system overactivity is that we have muscle tension that is pretty extreme, that's pretty pathologic or damaging, and there's dysfunction in the myofascial tissue. And when I say myofascial tissue, if you look at the diagram here, this is a muscle cell that's surrounded by a sheath of connective tissue, and then each of these muscle cells are bundled, and that bundle is surrounded by connective tissue. And then you can see that the bundle of bundles is connected by its own coating of connective tissue. And then finally, the whole muscle is encoded with what we call deep fascia. And so you can see that a muscle is not really just muscle cells. It's actually the muscle and the surrounding connective tissue that penetrates the muscles, and we call that the myofascial tissue. And it almost looks like a honeycomb, or it kind of looks like a slice of an orange. And it's really important to know that the muscles and the fascia are completely intertwined. They cannot really be separated, and that this seems to be an area in fibromyalgia where there is a lot of abnormalities. And it's important to kind of understand that. Studies have shown that there's excess levels of inflammation, that there's abnormal amounts of collagen, which is one of the building blocks of connective tissue, that we see that in the fascia surrounding fibromyalgia muscles in a way that you don't see it in healthy control muscles. So, the real message here is that short-term activation of the sympathetic nervous system is good, helps us contract our muscles so that we can fight or run away. But when it happens chronically, like it does in fibromyalgia, it can cause a lot of harm to the muscles. In 2010, I had an article published called Fascia, a missing link in our understanding of the pathology of fibromyalgia. And in that article, I outlined my theory that in fibromyalgia, the most of the symptoms, fatigue, fog, muscle pain, could be considered as happening in a chain reaction that starts with sympathetic nervous system hyperactivity. And if you think about it, a fight or flight nervous system that's constantly sending out the danger alarm is not going to enable you to get good quality or deep sleep, and poor sleep will lead to fatigue and brain fog. From the pain perspective, though, what we see is the sympathetic nervous system hyperactivity leads to the pathologic muscle tension I mentioned earlier, fascial inflammation, and muscles that tend to be riddled with painful knots called trigger points. So even back then when I was, writing that article, we knew that this process sort of ended up causing central sensitization. But what we didn't know was how it got from this step to central sensitization. There was kind of a question mark here. And it seems like this mouse antibody study fills in that missing puzzle piece, and that antibodies and the immune system may actually be what caused this final step, kind of connecting the muscle tension, myofascial inflammation and pain, trigger points to central sensitization. So this is a pretty important study, actually, and you might have heard about it. It was pretty big news when it was released in summer of 2021. And part of why it was big news was it was another objective or measurable abnormality that we could see in fibromyalgia. That each time an abnormality is shown in a research study, it further convinces people that fibromyalgia is real. It also brought up the really big question of, is fibromyalgia actually an autoimmune disease? And we'll go through what the study shows, and then at the end of the video, I'm going to give you my opinion as to what I think as far as, is fibromyalgia an autoimmune condition? So in this study, they infused antibodies from fibromyalgia subjects into healthy mice. And these mice that received those antibodies developed signs of pain amplification or central sensitization. So what they did in this study was they drew blood from fibromyalgia subjects and from healthy control subjects, and they purified out their antibodies and then injected their antibodies into healthy mice. And what they found was when they injected the antibodies from healthy subjects, nothing happened to the mice. Fibromyalgia antibodies, however, caused a variety of effects. I'm only going to focus on one that's related to kind of our talk today, and that is they noticed the induction of pain-like behavior, and in particular, they noticed that these mice seemed to be more sensitive to pain and things like cold. So it was like their nervous system was amplifying pain signals, and essentially they were able to induce central sensitization in these mice. So to kind of really understand what this means, we need to go a little bit back into understanding kind of how antibodies work in the immune system. And, prior to COVID-19, I don't think that people had as good of understanding. I think a lot of us now have had to become kind of more immune system and virus experts just to kind of understand what the heck was happening in COVID, particularly in the early days. But what we know is antibodies are proteins created by the immune system that float around the human body, waiting until they find their matching antigen. And so the analogy that's usually given is an antibody is like a lock, and it's floating around until it meets the key that fits it. And then once a key that fits it kind of activates or turns that lock, then a signal is sent to the rest of the immune system to come in and, help as needed. So we can understand really that antibody response can be protective. For example, if we have antibodies to a cold virus and then we're exposed to that virus, the antibodies are going to glom onto that virus quickly, right? They're kind of the first responders of the immune system, and then send out signals like, hey, immune system, activity needed here to defeat this virus. But antibodies can also be harmful, and that's when they become autoantibodies, or antibodies that are directed against self. And if you think about an autoimmune disease like rheumatoid arthritis, for example, we know in that illness that there are antibodies that develop against components of the joint tissue. So these antibodies are actually going into the joint and seeing keys from our own tissue and bringing in the other, kind of immune response. Hey, danger here. We need you to activate. And it's actually the activation of the other immune cells triggered by the recognition of those antibodies of our own cells as being foreign. The immune system comes in, attacks the joint, and that's actually what causes the joint pain and inflammation and actual joint destruction that we see in rheumatoid arthritis. So the researchers wanted to kind of know, well, hey, is this an autoimmune attack on the muscles, on, some other part of the body? So they sacrificed some of these mice, and that's a nice term that they use in medical research to say they killed these mice. We thank them for their service, however, and they looked to see where these antibodies were binding to. What antigens were attracting these antibodies, or what keys were triggering these locks. And what they found was the fibromyalgia antibodies bound to primarily a component of the nervous system called the dorsal root ganglion. And it's not really a surprise because the dorsal root ganglion is well known to be an intersection of the immune system and the nervous system, and it actually plays a pretty key role in developing central sensitization. So let's step into anatomy land here for a minute and kind of try to understand more the role of the dorsal root ganglia and what the heck they are. So dorsal root ganglia are these little peanut-sized collections of nerve cell bodies that live just outside the spinal cord on each different spinal cord level. So they kind of line both sides of the spinal cord. And they are actually filled with the cell bodies, or really kind of the brain of these sensory nerves that, the one arm of these sensory nerves exists out in the periphery, let's say, in your hand, where they are, they innervate both the skin and the joints and the muscles. And each of these sensory nerves, a signal can get sent up them, and then it actually goes into the dorsal root ganglia to be processed and then relayed to the spinal cord, and then the spinal cord sends the signal up to the brain, and the brain is actually what interprets the pain sensation, right? So one way to think about these sensory nerves is I like to think about them as little octopi that have kind of the head, or the brain of the octopus would be that cell body that exists in the dorsal root ganglia. And then these octopi only have two tentacles, one that stretches all the way out into the periphery, and then a much shorter one that connects to the spinal cord. And so, you know, if a signal goes into one tentacle, this long tentacle, it goes up, then processed by the brain of the nerve, and then that processed information is sent to the spinal cord. And the immune system is actually involved here as well, because we know that each of these cell bodies of the nerves are actually coded by immune cells. And so one of the inputs that help the dorsal root ganglia nerves know, okay, I need to amplify or not this signal, is what messages they're getting from the immune system. And certain immune signals trigger these neurons to become hyperexcitable. So it may be a normal signal that gets sent up here, but if the brains of these nerves are getting the right messages from the immune system or in other ways, they then send out a really loud or kind of hyperexcited signal to the spinal cord. And then the spinal cord says, got to turn the volume up on this one. And that actually right there, that's sort of the step that we would call central sensitization, because the signal coming into the nervous system is of a normal volume, but here it gets ramped up, and then the spinal cord is getting the message, oh, I need to actually turn up the volume even more on this signal to make sure the brain really hears it. And that is definitely, sounds familiar to everybody with fibromyalgia, I'm sure. And it's why something like a cat jumping on our lap can feel painful to us, where it's not going to bother somebody without fibromyalgia. So now back to the mouse antibody study, and there is the reference. The authors of this study, based on the information that they gathered, that the fibromyalgia antibodies triggered central sensitization and that they seem to bind most heavily in the dorsal root ganglion, what they theorized, or what they kind of concluded from this, is that perhaps these fibromyalgia antibodies are actually autoantibodies that are directed against the nerve cells that live in the dorsal root ganglia. So an autoimmune reaction against that part of the nervous system, and that this autoimmune reaction there sensitizes them and makes them send out the signal, hey, hyperexcitable signal sent to the spinal cord, ramp up the signal to the brain. And as I read this study many times and then thought about it for several months, I really had the gut feeling that their interpretation of their findings was not correct. And there's a few reasons for that. The first is, if we conclude that this is an autoimmune reaction against the dorsal root ganglia, you would expect a few things. You would expect that if they looked at where healthy subjects' antibodies bound, if it was an autoimmune reaction only generated by fibromyalgia antibodies, you wouldn't expect to see a lot of binding of other antibodies, healthy subjects' antibodies. But guess what? They actually found, when they looked at the mice that had received the fibromyalgia antibodies and those that had received antibodies from healthy subjects, both of them had a huge amount of antibody binding at the dorsal root ganglia. So that kind of doesn't make sense that we could say there's something different happening in the fibromyalgia antibody binding. The other big reason, to my mind, particularly because I'm so interested, and I think there's so much data supporting myofascial abnormalities in fibromyalgia, is that if we are thinking it's just autoantibodies against a part of the nervous system, how do we explain all those myofascial abnormalities? And in particular, how do we explain the benefit, the pain reduction that people see when they get their myofascial tissue treated, whether it's with myofascial release, gentle movement, gentle stretching, trigger point injections? How can we explain that things that are directed at the myofascial tissue would have benefit for pain if all the central sensitivity and pain signaling was being amplified just at the DRG? So this issue hits close to home for me because when I got sick and I developed fibromyalgia in medical school, the number one thing that helped me get back to school, I took a year leave of absence to try to find treatments that helped me. The number one thing that helped reduce my pain was myofascial release. And it really brought my pain down from like an 8 out of 10 to a 2 out of 10, to a level that was manageable, could be in the background, and I could go back to medical school. So to me, if it's their explanation is that it's happening just as an autoantibody binding at the DRG, then the benefit that I receive from myofascial release and the studies showing that myofascial release is helpful wouldn't make sense, right? Because what does the DRG care that you're getting myofascial release if the whole process is being triggered by an antibody attacking the DRG? The other kind of big gap I think that they're neglecting in their theory here is, how do we explain that central sensitization is seen in lots of conditions? It's actually seen in pain conditions as diverse as endometriosis, chronic low back pain, rheumatoid arthritis, osteoarthritis, or wear and tear arthritis. And in fact, there was a very interesting study done where they looked at people with bad hip and knee osteoarthritis, and they found that they had central sensitization. But then once that problem tissue was resolved by a joint replacement, so once there was no longer that kind of painful input, the central sensitization went away. So it's clear from that, to me, that there has to be something in the periphery, something in the body tissue itself that is triggering this reaction of central sensitization. But we never really knew the mechanism. So what I do think that this mouse antibody study showed us is the mechanism. And I think what all of those conditions have in common is that there's some low-grade or even high-grade amount of tissue dysfunction or tissue inflammation or tissue damage. You know, we know in rheumatoid arthritis, the joints are actually really getting damaged. And we know that in fibromyalgia, there's clear damage happening in the myofascial system. And I'll talk more about that in the next slide. But it wouldn't make sense to me if central sensitization, like we see in fibromyalgia, is caused by autoantibodies to nerve tissue in the DRG. It wouldn't make sense that there's all these other conditions that also experience central sensitization. It's more likely that this process at the DRG is sort of the common end result of all of these painful chronic conditions, and that the immune system is involved in that trigger, telling the DRG nerves to amplify their signals and telling the DRG nerves to also send chemical signals that cause the spinal cord neurons to say, whoa, gotta ramp up the pain volume. So I pondered for a long time what the mouse antibody study really showed us. And then I did a huge amount of research, and I read hundreds of research articles trying to kind of figure out, well, how can we connect what we know that's going on in the fascial tissue to this end result of central sensitization, but now including that there's this step that has to happen that occurs at the DRG. And this kind of chain reaction that I outline here is really the visual depiction of the hypothesis that I just had published in this article. And I will put a link in the description below so you can download it. You can actually download it free until July 26, 2023. After that, there'll be a charge to download the full article, but you can certainly always read the abstract or the kind of summary for free. So what we've talked about thus far is really kind of this line of the chain reaction, in that we know that there's sympathetic nervous system hyperactivity happening in fibromyalgia that's really driving a lot of this. And we know that this excess sympathetic or fight or flight nervous system activity leads to increased muscle tension, to the point that it's almost pathologic muscle tension. And there was a really interesting study that was done pretty recently where they looked at the different pressure in fibromyalgia muscles compared to muscles of healthy controls. And they did this by inserting kind of small pressure gauge needles, similar to what you use to check the air pressure in your tire. And what they found was pretty normal levels of pressure in healthy subjects, but fibromyalgia subject muscles had extreme elevations in their intramuscular pressure, almost to the level of concern for compartment syndrome. And what compartment syndrome is it's a medical surgical emergency where a compartment of your arm or leg has such swelling inside it that it's increased pressure to the point that you're going to get tissue death. And so they have to go in and release that pressure, or you're going to lose some of the tissue. Fibromyalgia muscles weren't quite that bad, but they were pretty darn close. And to me, it would make sense that because we have, unlike this extreme compartment syndrome level of pressure where you get extreme tissue damage, what we see in fibromyalgia is a more mild form of tissue damage. And that leads to, I think, a lot of the findings we've seen as far as fascial inflammation, as far as why we develop trigger points, why it's our muscles that primarily hurt in fibromyalgia. And so we've kind of known this part for a while. And this is really what I described in my 2010 article. The update 13 years later that I'm publishing now is what happens on this end of things. And so, the mouse antibody study really gave us the information that it actually takes an immune response or an antibody involvement to get us from here to central sensitization. So one of the kind of areas, there's kind of two things that I found out in my deep dive into medical research that kind of gave me the aha feeling of like, I think I might have figured this out. The first is there's actually been a lot of study of antibodies and how they cause or can cause development of central sensitization. And they've looked at this in various different animals and various different disease models, different antibodies. But consistently, what all these other studies found is that if you just apply an antibody to the DRG, nothing happens. Same thing as what they found in the fibromyalgia antibody study, right? Once they took the DRG out and they were just looking at what happens when you apply fibromyalgia antibodies, nothing. It took something that was happening in vivo or in the body to make that activation happen. And what these other studies found is antibodies themselves don't do it. What does it is an immune complex, and that is antibody plus antigen, or lock filled with key. So if immune complexes, if the DRG is exposed to immune complexes, whether in vivo in the body or in vitro, in a lab, that's what triggers them to become hyperexcitable and send that message to the spinal cord to ramp up pain volume, leading to central sensitization. And it kind of makes sense. The more I thought about it, I was like, well, why would it be that it's the immune complex that would trigger that? And then I realized, well, the DRG and the central nervous system doesn't really care that there's antibodies floating around. I mean, there's always antibodies. There's millions of different antibodies. The central nervous system does care if there is tissue damage, because that would be an appropriate time to actually generate pain hypersensitivity. And think about it like this. If, let's say we sprained our ankle, not only hurt a little bit, probably not going to stay off that and allow healing. But if we sprain our ankle and that generates pain hypersensitivity, boy, we're going to really baby that ankle. We're going to really stay off of it. And, that's why, if you think about it, that's why something, when you've, hurt yourself or sprained your ankle, even just like something with the blankets laying on it can cause pain. Well, that's because your central nervous system has become sensitized. And it's become sensitized because it has been exposed to the combination of an antigen derived from tissue damage. So, let's say cells from the damaged tendon. Those are floating around in the bloodstream, and then autoantibodies that we have at baseline, I'll go into that in a minute, are binding to it, and then that immune complex is floating throughout the circulation. Then it's filtered through the DRG, and there's immune cells in the DRG that would be like, wait a second, that looks like tissue damage. And if that's happening a lot, if the DRG is really being overwhelmed by those signals, central sensitization. So remember I said that autoantibodies can be bad? Well, they're only bad when they are abnormally created or they're in excessive levels. Actually, normal, healthy individuals, we all have some low level of autoantibodies floating around to all sorts of different body tissues, because these autoantibodies are acting as kind of security guards, paroling throughout the bloodstream and the circulation in search of either foreign invaders or signs of tissue damage. And let's say this autoantibody is floating around. In the case of the ankle sprain, it hits some of the tendon proteins that were released in this injury. Then they bind, right? And not only does that float to the DRG and cause central sensitization, that immune complex, that binding of the key into its antibody, sends a huge amount of other signals out to the immune system, recruiting other immune cells to the area. So, for example, autoantibodies might hit tendon bits of tissue that are floating around. They also might bind to the area of injury in the tendon itself and then say, hey, immune system cells, come on in. We need a cleanup crew and we need some help rebuilding. And then the cleanup crew is attracted there. That's usually the macrophages. They eat up damaged tissue, and then the immune system is also really involved in directing kind of tissue repair. So, it would make sense to me that in fibromyalgia, just like all these other studies that were found, that it was really an immune complex that was required to activate the DRG to cause central sensitization. It would make sense that the same process would be happening in fibromyalgia. And, if you think about if your muscles are really, really tense, even just normal daily activities are going to generate some injury, kind of a low-grade injury and a kind of a chronic low-grade tissue damage and chronic activation and pulling in these autoantibodies to bind to the damaged cells, the damaged areas, and then floating around up to the DRG and generating central sensitization. What seems to happen in fibromyalgia, though, is that these autoantibodies have also gone a bit haywire, and they seem to be generated in higher levels than normal. And I was thinking, I wonder why that is. And then as I did some research, what I learned is, guess what part of the body gives these autoantibodies kind of the stand down notice? Because, we don't want the autoantibodies to continue to bind to damaged tissue. Once the tissue is healing, we want those autoantibodies to go away so that they're not pulling in kind of that cleanup crew that could continually eat up tissue that's healthy. So we only want them there when there's damaged tissue. And the area of the body that controls and gives the autoantibodies the signal to stand down? Sympathetic nervous system, our old friend. So when the sympathetic nervous system is hyperactive, like in fibromyalgia, these autoantibodies don't get the correct stand down notice. So that means that they are kind of being ramped up. So they're being ramped up at the same time we have kind of constant generation of myofascial antigens or evidence of myofascial tissue damage. And so you have a whole bunch of those combining, making immune complexes that bind to the DRG. And what this means really is that the sympathetic nervous system is kind of the driver of this whole process. And it makes sense if you think about, let's say, an injury that was sustained in combat. You would want as much autoantibodies to be rushing to the scene as first responders as possible until the threat of danger has passed. And combat is probably the situation in our modern era that generates the most strongest activation of the sympathetic nervous system, the fight or flight response. So we don't want the sympathetic nervous system in that kind of danger setting. We don't want it to give the stand down notice to the autoantibodies until the danger has passed, so that we can kind of hope to heal as much as we can while we're still exposed to danger, pull in as much of the immune response until we can get away from danger. Then the autoantibodies can recede and the tissue healing process can kind of go forward. So to review, what we see in fibromyalgia, sympathetic nervous system hyperactivity. That leads to elevated intramuscular pressure, or extreme muscle tension is another way to say that, which means that even normal daily activity is going to generate muscle pain, it's going to generate myofascial inflammation, and this kind of constant low-grade tissue damage is constantly generating antigens or keys that are derived from myofascial tissue. Then we know that the sympathetic nervous system also has this effect on autoantibodies and tissue healing. So basically, as long as the sympathetic nervous system is still giving the signal to ramp up autoantibody production, you're not going to get full tissue healing, because these autoantibodies are kind of saying like, hey, bring in the destruction, bring in the cleanup crew. And it's not until the autoantibodies kind of back off that the real healing or kind of construction of new tissue can occur. But because the sympathetic nervous system is constantly driving this process, we see impaired tissue healing in fibromyalgia, persistent autoantibodies, and the autoantibodies most likely are to the tissue that's being damaged. And that, in my hypothesis, is the myofascial tissue. So we have then a huge amount of keys and a huge amount of locks floating around that combine together and then send the message at the DRG up, tissue damage happening, gotta ramp up central sensitization. So I promised at the beginning that I would give my 2 cents in the last little minute here as to whether we can consider fibromyalgia an autoimmune disease. And I think what this means is that we can consider fibromyalgia a partly autoimmune disease, in that there is abnormal activation of these autoantibodies, and that is driving the process of central sensitization. But we also need to remember that fibromyalgia is a disease of the myofascial tissue and of the sympathetic nervous system. So that's why fibromyalgia is so complex. 3 different systems in which things aren't working correctly. So I would say partly an autoimmune disease. The good news about that is if we understand the role of the immune system in fibromyalgia, that means we can really expand our treatments. It means that all of those kind of autoimmune disease treatments that are used in other autoimmune conditions like rheumatoid arthritis or lupus could potentially be added to our treatment tools. And I will tell you that there's a huge amount of research going on now, because every drug company out there that makes anything that treats autoimmune disease is now looking at their drug to see if it helps in fibromyalgia. So stay tuned. I'm hoping that we'll get some good news out of that and have added some new treatments. I also will say, though, we need to not forget that we still will need to treat the myofascial dysfunction and the sympathetic nervous system dysfunction to really get significant improvement in fibromyalgia. So in my book, The Fibro Manual, I go much more in depth about myofascial release and other treatments directed at the myofascial dysfunction in fibromyalgia. I go into great detail about treatments directed at sympathetic nervous system dysfunction in fibromyalgia. And there are some treatments that I discuss in that called primarily low-dose naltrexone, some other kind of immune modulation things that also can affect this immune system issue that we've now discovered is happening in fibromyalgia. So remember, you can download a copy of my article in which I go over this hypothesis in detail for free until July 26th. Until we meet again, stay fierce.